Good afternoon, everyone. Well, I'm really excited today because it's a presentation that I've been waiting for several months since we started to talk about this N3C Data Enclave. And we are very lucky because we have two speakers today, Suzanne McCahan and Stephane Meystre, who will present everything we know about the new COVID registry, everything we need to know, funded by NIH, this N3C Data Enclave. But before I introduce our speakers, let me go over some housekeeping issues. So as usual, we will provide CME credits for our tech talks and our ID series presentation. And we will distribute the information via email to those that registered for today's event. But if you did not register prior to today's talk, you can still obtain CME code information from Lisa Maturo, at Questionnaire, and I will enter her email into the chat box. The schedule for the 2021 ID series and tech talks will be coming soon, so stay tuned. So again, I'm really excited to present our speakers and to hear about the N3C registry. So Suzanne McCahan is Assistant Research Scientist at Nemours Biomedical Research. And as such, she supports and mentors investigators in the use of PedsNet, which is a large national pediatric database governed by a consortium of institutions, of which Nemours is a very active member. She also mentors and teaches people on how to use REDCap and now how to use the N3C Data Enclave. She obtained a PhD in molecular biology at Princeton, and is also a very active member of the BIRD, our Biostatistics Epidemiology Research Design Core of ACCEL. So thank you so much, Suzanne. And then Stephane Meystre is professor and chair in translational biomedical informatics at MUSC. He obtained an MD from University of Lausanne in Switzerland and a PhD from the Department of Medical Informatics at University of Utah. He has been funded by multiple federal grants, mostly focusing on making unstructured clinical data accessible for research and clinical practice by applying natural language processing approaches to extract information from clinical narrative text and protect patient privacy. I think, Dr. Meystre, you will have to come back to talk to us because that's something we are also very interested in at Questionnaire. He's had numerous publications, and he also leads a group that the N3C team group at MUSC. So thank you very much for our speakers, and we look forward to your presentations.
Hey, this is Suzanne, and I am going to start off. Okay, I should be sharing my screen slides now. Can you see them, Claudine?
Yes, perfect.
Okay, good. We are going to talk about the NC3 Data Enclave, which is a powerful national resource for COVID-19 research. I'm going to give an overview of the NC3 Data Enclave, and then Stephane is going to talk more about the data that's within the enclave and how it gets there. So N3C stands for the National COVID Cohort Collaborative. The NC3 is a partnership with overall stewardship by the NIH National Center for Advancing Translational Sciences. The National Center for Data to Health has a large role in NC3. The CTSA, the Clinical and Translational Science Award Hubs, also are contributing a lot to the NC3. There is a network of CTR programs that are contributing, and others, some additional healthcare organizations and research communities are contributing. The NC3 CTR network consists of 8 CTRs, and Delaware is one of them. This is led by Dr. Sally Hodder at the University of West Virginia. The NC3 network, the CTR network, is providing data from rural and urban centers of IDEA states. And this work is supported by a grant from the National Institute of General Medical Sciences. The N3C Data Enclave consists of a large health record repository. More than 72 sites are planning to provide data, and that includes Nemours. Records of patients who are tested for the novel coronavirus or have had related symptoms are going to be included in this repository. The Enclave also includes some powerful analytics capabilities. There are tools for online discovery, visualization, and collaboration. And specific tools include R, Python, SQL, dashboards, public data sets, and more is going to be added as time goes by. This is all available to DE-CTR investigators free of charge. There are N3C Domain Teams. These teams enable researchers to collaborate in a team science environment. The teams are comprised of clinical and subject matter experts, statisticians, informaticists, and machine learning specialists. And this is complex data with complex questions that are being asked and attempted to be answered. And so it's going to take a variety of expertise to use this resource effectively. And the teams that have already been set up are focusing on acute kidney injury, diabetes and obesity, pediatrics, in the immunosuppressed or compromised, social determinants of health, and many others. These are just examples of what teams have already been set up. And teams are continuing to be developed, and we encourage everybody to join a team or to start a new one. There are N3C projects already designed. There are about 80 that have already been proposed and approved. General information about the approved projects is shared with the public. The N3C Data Enclave users can request access to a project and contribute to that project. Some of the projects, I've listed some examples of the projects that exist. And some of them are acute kidney injury in pediatric COVID-19 patients. Another one is examining associations between vitamin D status and COVID-19 test results. Another project focuses on patients with diabetes, and others on health disparities. So that there are many projects. I mentioned about over 80 projects. And you can join a project or propose a new one. There are 3 tiers of data access for the N3C. The Tier 1 includes synthetic data. This is artificial data that's designed to be statistically comparable to the original data. It doesn't contain any PHI. Tier 2 is de-identified data. This is patient data. This is real patient data, but it doesn't have any PHI identifiers. And then the third tier of access is a limited data set. This is real patient data, and it contains 2 elements that are defined as PHI. The requirements for access differs for each tour, and DE-CTR investigators are eligible to request use of any tier of data. There are several things that are required when you request access to the N3C Data Enclave. An institutional data use agreement is required, and each of the DE-CTR institutions has a DUA in place already. So part of that work is done. Everybody who requests access needs to have a personal ORCID ID. This ORCID ID serves as a unique identifier for everybody, and it allows for tracking of what's done in contributions that are done within Enclave, and then everybody's contributions can be acknowledged appropriately. A 2-factor authentication app is required, and there are several options to use. Current human subjects research training is required, but this doesn't apply to this access to the synthetic tier of data. And current NIH information security management training is required. There is a specific website and course that's needed, and that takes about an hour to complete. Project information is required, the name of a project that's been already approved, or a new one can be proposed, and a IRB letter of determination from your institution is required if you want access to Tier 3, the limited PHI access of data. So the information that's needed when proposing a new research project is the title for the project description, and the project must be COVID-19 related. And an abstract and rationale are required, and the rationale should include what data tier access is requested and a justification for why that level of access is needed. Collaborators can be listed on a proposal, and that would require their name, institution, and email address. The collaborators do not have to be from the same institution as the lead investigator, although each institution must have a DUA in place. The lead investigator's name, project title, and project abstract of approved projects will be made public. And I've listed some URLs, links for more information and for help. DE-CTR members can put in a BIRD request for help, and this is the link for the BIRD request. And you can request help for obtaining access to the NC3 Data Enclave. We can help you formulate a data use request, and we can help you use the Data Enclave itself. We have created some NC3 web pages that contain a simple outline and links to more information. And I've also listed the website for NCATS for N3C and the N3C website itself. And that is the end of my part of the presentation. I'm going to let Stephane share his slides and tell us more about the Enclave.
All right, thank you, Suzanne. Let's see. You see my slides?
Yes.
Yes.
All right, perfect. Yeah, thank you for this opportunity. So what I'm planning to tell you about is a bit more details about the National COVID Cohort Collaborative, N3C, more specifically about the side of things where data is being pulled out of existing electronic health record systems, data warehouses, etc. So from healthcare institutions and prepared for sharing with N3C and then combined and made available in the Enclave. And I'll also tell you a little bit about what we have done at MUSC around these efforts, what we have done and we are currently doing. So very briefly, I mean, you probably all or obviously all know about that, but the efforts that I would start telling you about started in about mid-March, very close to when the World Health Organization declared COVID-19 a pandemic. And so we were observing quick progression in South Carolina and other places, New York and so on, but no real idea of what would be happening and that it would happen this way, higher and higher. We had very fast growing cases in South Carolina and found a way to try and manage patients worrying about COVID-19 or wanting to be tested by using a telehealth application to offer virtual care visits, a product from a company called Hypnosis. And so it's a system where patients were answering questions, do you have the atrial congestion? Yes, no, cough, yes, no, etc. And then the system was exporting some kind of notes generated from this data that we then integrated into the electronic health record system. So we were accumulating clinical information from patients tested for SARS-CoV-2 with COVID-19 diagnosis sometimes, and had an urgent need to be able to use this information first locally and then wanted to share it more broadly. So locally, we had this stored in the electronic health record system. Chronicles is the transactional database, so the main one where all the information is stored. And we started this effort on March 16, and then added also other sources of information from the system, and then established a dedicated data mart at MUSC, going through our data warehouse and other resources, and then also developed a new natural language processing application to be able to have the structured and coded data out of these interactions with the patient, so that we could know all the symptoms the patients were presenting with, the treatments they might have taken, the investigations they might have had already in the past, etc. To extract all this information and store it back into a data mart that was partly using the standard data model that N3C is using, and I'll tell you more about that. And so it includes information from these nodes from the virtual care visits, but also everything else. Well, not everything, but a lot of the relevant clinical information we had at MUSC for those patients, from diagnosis, procedures, all lab tests, including SARS-CoV-2 diagnostic testing, history of their visits, etc., and combine this all with the information we got from these interactions, virtual care visits, and could also link that with the results of the testing. And so I'll tell you later how it can be useful. So it was first, as I mentioned, for supporting healthcare operations locally, also a very strong interest for research developed rapidly, and this data mart that I mentioned was used to answer both of these needs with this information, dashboards, etc., that the clinical side of operations were using, and IRB established processes to give access to this new resource, data mart, and also plans to share it locally, so nationally. And so we started sharing, working with N3C very early in the process. I think MUSC was among the 10 first institutions establishing all the legal requirements to share data with N3C, among many other ones. And so now I'll tell you more about N3C. There will be a little overlap with what Suzanne has said, but I'll insist more again on the kind of data you can find in this resource, which patients are there, and how this information is represented, how you could be able to use it, and how it ends in this enclave for use afterwards. So a little more about the N3C, the National COVID Core Collaborative. It's organized in work streams, so groups of experts who are focusing on different aspects of this effort, starting with governance, etc. The phenotype and data acquisition is one that I will mention more, and then data injection and harmonization. So it starts from the institutions sharing, and then it's ingested and harmonized centrally, and then collaborative analytics helps developing resources, interfaces, and tools for researchers to access and analyze the data. And synthetic data is a separate one that Susan mentioned, so I'll tell you a little more about that also. So currently, well, this was like a few days ago, 44 sites had shared data with N3C already, so had deposited data in the N3C pipeline. And they were using different data models. I'll tell you more about those, but it's between 14 who use pCoreNet and 7 who use ACT. So there's a variety. You see that there are sites in many different states, more on the east, but this is really rapidly growing. There are more than 70 who are in the process of establishing all the legal requirements to also share their data. 73 as of December 16. So this is institutions sharing their data, and then 134 have a data use agreement. So this is institutions wanting to use the data, access it for research applications. And as of December 16, the N3C Enclave includes information from 2.6 million patients, and 373,000 approximately of those patients have positive SARS-CoV-2 diagnostic tests. So they are COVID-19 positive patients. And it includes hundreds of millions of different types of information. I'll tell you what these observations or procedures correspond to, but it's about 1 billion lab results and 400 million medications, etc. So it's a large data set, a large database, and it's growing fast also. So it will grow larger pretty rapidly. Now, the process from the source of the data to use for research is shown here, very high level. So it starts on the left with the institution sharing the data set using one of those data models, ACT, Trinetics, Picoria, or OMAP. I'll tell you a little more about those. And then the ingestion process to then harmonize it into one common data model called OMAP. That's the Observational Medical Outcomes Partnership data model. I'll tell you more about this one. And then from this harmonized database, then generating a de-identified data set, a limited data set, and synthetic data also that is by definition fully anonymous, and then making it available for researchers to use. So participating institutions, as I was mentioning, have to use data stored in databases that use 4 different data models to make it possible for as large group of institutions as possible to share their data. The plan for N3C has always been to accommodate different data models, and so they chose the ones that are known to be the most successful and prevalent. Odyssey is the observational health data science and information and informatics effort that continues developing resources that were developed initially in the OMAP efforts and uses the standard data model that I'll tell you more about. PCORNet, it's the Patient-Centered Outcomes Research Network. You might have heard of it. So it's also a popular one. The Accrual to Clinical Trials, the ACT network, uses another data model from an earlier effort called I2B2. And then Trinetics is a commercial company that also has its own data model. So institutions who have clinical data in databases that use any one of these 4 data models can participate and share the data with N3C. And so this is done or eased by the availability of scripts that N3C develops and makes available that make the work of selecting the patients, so querying those databases that use one of these 4 data models, querying them, preparing the data, packaging that, very easy, and also makes it consistent, so done exactly the same way for each institution participating, whatever the data model they use. And then these institutions share all this information in some large archive file that is sent securely through some encrypted SFTP data sharing with N3C. And then N3C goes through a very complex data verification, mapping, and harmonization process to have all this data stored in their standard data model that's based on the OMAP CDM, and I'll tell you more about that. So this is how the data that is available in the enclave is represented. Now, very briefly about this process that N3C goes to from receiving data from different institutions using different data sets to centralizing it and making it available in the enclave. So they start with basically restoring the data that was in this archive into a new database that reflects or mirrors what the sharing institution had, and then do all sorts of verification, looking for missing information, validating the formats, checking the validity of the codes, of the dates, of everything, and then do verifications and checks all the line to make sure that the data is as complete and correct as possible. Then they follow with terminology mapping with a tool from a company called Adaptiva. So this is mapping the different terminologies that the institutions used in those databases that use one of these 4 standard data models to the standard terminologies needed to be used in an OMAP CDM database and available in the enclave. I'll tell you more about those, what these standard terminologies are. It's basically mapping between those and then staging this information again in a new database that this time uses the OMAP CDM data model, merging it all and then exporting that to the platform that the enclave uses that resides in the cloud. So in a very secure, certified cloud and platform developed by Palantir for the NIH and other federal institutions. Now, the patients that are being selected, imported, and then shared with N3C have to follow specific criteria. And these criteria are defined in the N3C Phenotype that was first defined in April 2020 at the very beginning of the project, including data for those patients selected starting January 1st, 2020. And so the first version of the phenotype, again starting in April 2020, included pretty much anyone tested for SARS-CoV-2 or who had a diagnosis of COVID-19 or anything related, a weak diagnosis code or something that made it possible that the patient had COVID-19. So there are 4 categories of patients: lab confirmed positive, that's pretty clear. So a list of diagnosis of lab tests and their codes is made available to verify this, and only select those who are in general about SARS-CoV-2 diagnostic testing. Lab confirmed negative, the same test but a negative result. The suspected positive, so these are patients who have a diagnosis that specifically is for COVID-19, and a few have been created as emergency codes by the World Health Organization, for example, for the ICD-10 terminology and other ones. So these are used in this category. And then finally, there's a long list of possible positive patients who have diagnosis codes that are related, like the example I have here, an other specified respiratory disorder. So it could be something for pneumonia without clear cause, or paragusia, or all sorts of possibly related observations. So you can imagine that these categories, the lab confirmed negative, growing progressively with the testing becoming more and more available and many people testing, and for the vast majority having negative results, but also these possible positives made these cohorts of patients selected for N3C grow very fast, too large. And something had to be done to not overload completely this process and the databases. And so the second version of the phenotype was published in August, and it changed a little bit how the patients are included in these categories. So the lab confirmed positive, negative remain the same, except that a few codes have been added. So new products for testing, diagnostic testing, etc. were added to the list, but by definition they are about the same. So they had a diagnostic test that was either positive or negative. But then among the suspected positive, and more specifically also the possible positive, the criteria were made a little stricter. So for possible positive, they had to have at least 2 of these weak diagnosis codes. And the main difference also was that every case who has a screening code, that's a code that all institutions or most institutions were using when people were presenting for SARS-CoV-2 testing without clear diagnosis, just for screening, and had a negative test. So these were lab confirmed negative in the phenotype version 1, but the second one, they were excluded if this was not followed by a positive SARS-CoV test or some suspected positive or strong diagnosis. So the goal here was to drastically reduce the number of negative cases and make it feasible to accommodate all this data sharing in cohorts of patients. But it still included a lot of negative patients, but fixed some of the problems that were present. And then the third version was released less than a month ago in November, the end of November. And this time changes things a little bit again. So in this case, instead of removing all these negatives, as I mentioned before, which seemed to be too much, then they added criteria to establish control patients who are confirmed negative, but have some characteristics that match those who are confirmed positive, and are selected only in the 2-to-1 ratio, so that they are approximately twice as many negative patients than positive patients among the lab confirmed. And so this hopefully will allow balancing the population of patients that are being shared and stored in this enclave. Now, for those patients who have been selected in one of those 4 categories, either the lab confirmed positive or negative, the strong suspected or possible positive for COVID-19, then all this following data is being stored. Almost all of it. I'll tell you what's missing. But basically, it's representing a large variety of clinical information for those patients. And it includes, in this blue frame here, the clinical data itself that I'll tell you more about, but it also includes information about the health systems, economical cost information also, if this is shared by the institutions, but it's possible and the system can accommodate it. It has also standardized derived elements, cohorts of patients, for example. So these are groups of patients who correspond to specific criteria, and the ones that are, for example, created in the N3C projects that Suzanne mentioned before. And then you see a variety of standardized vocabulary. So this is one of the characteristics of this OMOP-CDM data model, the Odyssey standard, and what's being used in the N3C Enclave is that all the data is using a standard data model and standard terminology. So this makes it possible to combine the data from multiple different institutions and then readily use it for research applications. All the data is coded, structured, and coded in this database. Now, looking a little more at the clinical data tables to show you what's available. So there's information about the patient, obviously. This is somewhat limited by design. The database, the OMOP-CDM, and what's used in N3C is almost limited data sets already. There's no recording of the patient name, for example, their address, or anything like that. So the only information available about a patient or a subject is their date of birth and some demographic information. And then you have information about what's called an observation period. So these are periods when there's a possibility that information about a patient is being recorded in the system. It could be empty. If it's empty, it means that nothing happens with the patient, which is very useful to know. And if something is recorded, then it has to be within this period. No data can be recorded without being in an observation period. And so this corresponds to, for example, a time when a patient was being initially treated and then followed up for some problem, or can span years for some chronic disease being treated, etc. And then you have every interactions, visits, or encounters that are being stored. All the diagnostic therapeutic procedures, conditions correspond to diseases, but also signs and symptoms. The drug exposure corresponds to the medications, vaccines, and other biologics. Devices are for almost every object or instrument used for diagnostic or therapeutic purposes. Measurements include, for example, lab testing, anything that is systematic and standardized for examination or testing. And then everything that doesn't really fit in those relatively standard categories then is called an observation. This can include questionnaires, social history, family history, all sorts of also interesting information. The specimen are about samples. Death information is stored. This is recognized as relatively sparse currently and still a problem, but in theory it could be stored there. And then some relationships between facts can also be stored as a fact relationship. For example, indicating that a child is the child of a specific parent, that a mitigation is linked with an adverse drug event, etc. And then grayed out at the bottom here, you have tables called Note and Note NLP that have been created and exist and are made to accommodate clinical notes, free-text notes, as you find them in the electronic health record, from history and physical reports to progress notes to discharge summaries, anything. And then the Note NLP one is for all information that's being extracted from those notes. So these are ones that we use a lot locally, but centrally within N3C, they're not being used yet. So there are plans to make this possible, but it's still not happening and might not happen. So currently, there's nothing in those tables, nothing you can have that is not structured and coded and in the tables that you see the other ones here. And those tables, so this is maybe a little too much details, but as I was mentioning, all this information is then linked with concepts in standard terminologies. And so these records, these records in this concept table link this mention in the N3C Enclave, the OMOP CDM database, with standard terminologies. And it makes it possible to, for example, keep the source terminology concept and link it with the standard terminology concept that has to be used in OMOPs. You know, sometimes those mappings between different terminologies are not perfect, and being able to see the source, what was originally used, can be very useful. So that's also possible in this database. And the standard terminologies used are several for different types of information. So you have the source concepts, the ones at the bottom here, who are the terminologies most frequently used in healthcare institutions. You know, ICD-10-CM, for example, historically ICD-9-CM, and many other ones. And most of those are then stored to the mapped to the terminologies that are considered standard for OMOP CDM and for N3C. For example, SNOMED CT for a lot of clinical information, and other ones that are helping with the classification to navigate between something that's very detailed to something that's far less detailed depending on the needs. So from low to high level terms, and MedRA is used for that, for example. So for the different types of information, here you have the standard terminologies. That's the second column. This is what you will find in the N3C Enclave. And then the source are what could be coming from other systems that were sharing their data. So for conditions, diseases, signs, symptoms, it's mostly SNOMED or ICDO for oncology. For procedures, it's also SNOMED and other ones. ICD-10 PCS is possible. For measurements, again, SNOMED and LOINC, that's for lab tests, and RX norm is for medications with some extension, mostly. Devices, observations, also SNOMED, and then for visits, other specialized ones. So this is just to say briefly that you will find a small selection of standard terminologies that code all this information in the N3C Enclave, and the possibility to have this link back to the source terminologies in the way that I mentioned before. Now, besides this clinical information, the existing one, there's also synthetic data. So this is a data set, as mentioned by Suzanne, that is being created using various algorithms that use the statistical characteristics of the population in the enclave, and then generate data, synthetic fake data, basically, that at the population level mimics the characteristics of the source population. So, for example, the proportion of females with a specific gender and ethnicity will be the same in the synthetic data sets than in the source real clinical data set. So this is useful for patient population level analysis and research, for hypothesis generation, but it's not usable for patient level applications. So every individual record has absolutely no individual validity. It could look reasonable and realistic, but it doesn't correspond to anything real. The advantage is that it's anonymous by design and therefore much easier to access. Now, among the data that I mentioned, there's also different categories of data that are currently missing in the N3C Enclave. And sometimes because the institutions rarely share them, or sometimes because the current version of the database does not accommodate them readily, or because the mapping and ingestion process, etc., does not include and map them completely or in a as accurate manner as possible. So this includes, for example, ICU admissions and status, ventilator use and use of oxygen, for example, administration of vaccines, even if it's in theory possible, as I mentioned before, with those terminologies. Many adverse events are not recorded. Family relationship records are also missing. EKGs, death status is very sparse, as I mentioned, as is the DNR status. And then details of ICU stays from the flow sheets, that some of the drip medications, infusions, and other types of information is missing. Convalescent plasma use and other blood products is also mostly missing. Payer information is mostly missing. And then admissions and visits in patient ones and other ones about pregnancy are also missing. So it's already extremely rich, but some types of information, especially those here, are missing. They are desired. There are many discussions and efforts to try and have them to enrich the database with this type of information, but it's currently not available or rarely available. Suzanne already mentioned clinical domain teams. So this is the exhaustive list of those. So these are groups, and this is a recent effort. So these groups are, for most of them, getting organized and starting activities now, or did so very recently. So it's really easy to join them if you have any interest in any of those, or create new ones. Okay, so now I'll tell you a little more details about the data access. I think Suzanne mentioned already some resources and information. I'll just give you a little more details and then also give some resources and links for more details for you to use. So, and I'll be brief so that we have time for some discussion. So again, you have the 3 tiers. Here you see again the process from ingestion to mapping, harmonization, etc., and the access. And I'll just tell you a little more about the different access levels, the 3 tiers. So the registered one accessing synthetic data is the easiest. The control one includes the de-identified data, but also aggregate counts. So if you have only access to aggregate counts without the data itself, that's also at the controlled level. And then the controlled plus is the limited data set. So that's the all, it's like de-identified data, but with full dates, full zip codes, and more detailed location information, basically, which might be useful. In a de-identified data set, all the dates are either removed or shifted, so you don't have link with other synchronous events happening. So for COVID-19, that might be a problem, and that's why this control plus, the limited data set, is made available. But it requires also more justification and is more difficult to access. The data use agreement is required for everything, human subjects training for everything except the registered one, the access to synthetic data. There's an IRB determination letter required for the control plus for the limited data set. And then for the de-identified one, some institutions require it, other ones don't. And the data use request, that's always required. And again, for the control plus the limited data set, you really need to justify why you need this level of identified or not completely de-identified data. So a rationale for accessing the data. And the approval goes through this data access committee, but also the IRB in that case. And so that's what it would look like. And I think I'll skip what we have done at MUSC and stop there.
Well, thank you so much, Suzanne and Stephane. Any question? Well, I have several, so I can start while everybody thinks about their questions. Stephane, how often are the data refreshed, and how often does every institution, provide data?
Yeah, excellent question. Forgot to mention that. So N3C asks for a weekly refresh at least, and they used to require it twice a week, but backed up a little bit also because they were somewhat overwhelmed with the data with the first version of the phenotype, as I mentioned before. But so now you can count on something that's weekly. from all these institutions. So it's really quite current.
And I mean, there is a ton of processing, as you described, at the N3C level. How long does it take for them to, so when they receive the data, to process all the data so that it's in the common data model and all the standardization is done?
So I don't know all the details to that. What I can tell you is approximation. So it varies a lot depending on the source data model, what data model the institution sharing data is using. So for those who use OMOP, for example, that's already the same data model. It's much easier and faster, and it's in the order of a few, maybe 1 week or a few weeks. But for other ones, they have to do more mapping, transformation, etc. That takes more time. And they had to build all these resources. All these pipelines had to be built, and the latest ones to go online and were making it possible to share data were only maybe a bit more than a month ago. So it took easily in the order of 6 months to establish that. So it's something that is still dynamically changing and will be faster, but data made available in the enclave might very well be like 1 month old after its recording initially. But that might be less in the future, yeah.
Here's a question. Let me check the chat. Don't see any other question. Well, I still have other questions. You mentioned that, Stephane, that the convalescent plasma was not right now in N3C. Is there a plan for it to be? And also, are the other treatments in the N3C?
Yes, they are. So most of them are recorded and shared. Now, I'm not aware of the details of each of these data elements missing and the details of the efforts to add those to the enclave. But all those I listed are recognized as missing, as being desired, and subject to efforts to add them. The problem is that what's being shared changes from institution to institution. Many of those institutions sharing, or almost all of them at the beginning, already had a database using one of those data models. But everyone was sharing it with slightly different types of data, and some data always missing. And so even if they progressively add or have the capability centrally for N3C to accommodate, so ingest and map and share the data if it's being shared, it's also missing from the sharing institutions. That's probably mostly the cost information, etc., for example.
Right.
This is Suzanne. A follow up comment about that and kind of a question. The convalescent plasma, I think that's an experimental procedure. It's not a standard procedure. Is that correct?
Well, I mean, I think it's under an EPA, but it's still provided, in multiple institutions.
Yeah, what's standard?
Most of the treatments are not standard procedures.
Yeah, that's true.
So the problem, like with remdesivir and other ones who have been, newly adopted, accepted for treatment COVID-19, is that the institutions have to establish a way to code this information, to collect it consistently and have it coded in one of those databases that can then be used to share the data with N3C. And coding medications like remdesivir, for example, was easier considering their existing workflows than other ones like convalescent plasma. And convalescent plasma, I'm trying to remember who, but some are sharing it. The main issue is that it's rarely done, even if it's very sparse what you will find in the enclave about that.
Yeah,
so
in terms of what's in the enclave, more standard kinds of treatments are more likely to be there. than something that's new, because it's just not in the medical record in a way that's easy to include it in the enclave.
Right. You could imagine something that is not coded, that is mentioned in multiple notes in the patient record in an institution, very broadly. If it's not being coded somewhat in some way and then exported in those databases used for sharing, you won't find it in the enclave. So the enclave is really, it's a wonderful resource in terms of size, quality, or cleanliness of the data and mapping between the different institutions, but it's only a portion of the information documented locally in an electronic health record about each patient, obviously, especially because it's lacking all what is found only in the text notes.
Right.
And so about that, there are efforts and a working group also within N3C, Natural Language Processing Working Group, working on strategies to make it possible for institutions interested in sharing data that either is extracted from those notes locally and then shared to N3C, or sharing the notes after the identification with N3C. So different strategies are being discussed to come up with something that would allow enriching this information significantly.
Yeah, that's amazing. So at MUSC, you are sending the text notes, right? It's part of your OMAP process.
No.
Oh no? Oh, okay.
No. So we have, so locally we have various databases that use the OMAP CDM. We are rebuilding or building a new one now that uses completely the whole OMAP CDM, including information extracted from notes. But the one we now have that I mentioned at the beginning, this dedicated data Mart, is like a hybrid. We didn't have time to build a fully standard one. So what comes out of the notes is using the OMAP CDM, but everything else is using the data model used in our research data warehouse. And then we use even another one for sharing with N3C using the ACT model. So it's a bit not optimal currently at MUSC, but we are working on a fully OMAP CDM based resource now. So that's in progress.
That's amazing.
I have a specific question about how the observation period is determined.
Yeah, that's a good question, and I'm not really sure. The problem is that it's a topic of discussion within the Odyssey community. Some do it a way, and others do it a different way. And I don't know exactly how they decided they would do it for N3C. So that's something that we could find out, I guess, but I'm not sure.
Yeah, because it's from what you said, the data that's included in the enclave is only within an observation period. So
yes,
I'm wondering if some of the data might be missing, if it's not, or something you might be looking for wouldn't be there because it's not in an observation period.
Yeah, but you know what's possible to avoid this problem and the diversity or the variation between the different institutions, and the fact that the other data models, the ACT, Trinetics, etc., don't have these requirements. Maybe in the enclave they just created an observation period starting on January 1st, 2020 for everyone, and that's it. That's possible. I'm not sure. Yeah.
So Suzanne, do you want to talk a little about the process that we are putting in place, with ACCEL and with you at Nemours to help or facilitate the access to the N3C database, for investigators?
Right. So if I posted the link for the BIRD request system, and those requests, if you put information about what it is you want help with, comes to the BIRD committee, and then we can have a group of experts who can help with the various levels of process. So it would be personalized help for whatever your specific question is.
Right. And I guess what we haven't mention also are the tools that are needed to be able to work on this data, on this platform. I don't know whether, Stephane, you want to take this also?
Yeah, so I didn't include that in the presentation, but they are, so the idea with the enclave is that the data stays there mostly. There are options that make the download of data a possibility. So like here, if you have a good justification for downloading synthetic data, you can. That's being in process. Aggregate counts, you can also download query results, but the data itself, so the real clinical data, de-identified or as limited data set, cannot be downloaded. So all the analysis has to be done in the enclave. And in the enclave, they make several tools available. It's mostly some that use the R libraries, some that use Python. And you also have the original Odyssey tools available. So the main one being Atlas. Atlas is a web application that allows querying this data. So you can build queries without having to know any, database language like SQL or something like that. So you can, search for specific concepts like a disease or demographics or age categories or anything, and then combine those with logic operators and or as well as temporal information, number of instances you want, etc. So really build your query and then see the corresponding counts that match and do some data analysis within it. And then also
Go ahead.
I was also mentioning, maybe this is what you were about to say, Suzanne, the projects that you mentioned. So several are creating those resources with characteristics of groups of patients, analysis done, etc., and making them available for sharing also within the enclave.
I was also going to say there are additional public data sets in there, like things that might be related to zip code, like
census data?
Excuse me?
Census data?
Yeah, maybe census data. I don't know specifically which data is there, but socio and economic status of different zip codes. And it all stays within the enclave. And if there's something that's not there, you can request it, and there's going to be a process to include that in the enclave so that it can be used in an analysis. And there's also the beginnings of some training on how to use the tools that are already in the enclave. That's being set up, and there is some training there available now. There is also some sharing of concept sets and phenotypes that are being defined, and those are being defined, verified, and approved to be shared with and then that would be a database that's available to be used, of different codes that are available to be used. So it's really meant to be a collaborative effort, and will probably require a collaborative effort to actually be used.
Exactly. Well, thank you so much. I think we are out of time. There are so many other questions that could be asked, but this was great. Thank you, everyone, and happy holidays to all of you. And thank you, Stephane and Suzanne, for this wonderful presentation.
You're welcome. Thanks for the opportunity, and happy holidays to everyone. And if you have specific questions, also don't hesitate to send me an email.
We will. Absolutely.
All right.
Thank you. Bye.
Bye-bye, everyone.
Thank you. Bye-bye.